Promising developments this morning that we want to get to in the race to find a vaccine for the coronavirus.
And to deploy it in every single corner of the world.
The world is waiting for a vaccine.
Scientists are at war with COVID-19. Dozens of laboratories are now battlegrounds in the race for a vaccine.
Today, we are making our third webiESOP. It's a very good thing for us because when we were starting these sessions, starting meetings, and tonight we are together again, not maybe face to face, but from by using internet and like these systems, we can find a way to come together, to share our thoughts, to share our experience, and to get knowledge from experts, our experts. Like our weekly webiESOP organizations, we will start at first with our president, lovely president, Klaus Meyer. Klaus Meyer will give information you about our last week results of COVID questionnaire. You know, we are publishing this questionnaire every week, and we are updating it, and we are releasing the results in our website. Actually, we collect lots of information and lots of results in these weeks. Sorry, 2 weeks later, the corona-infected pharmacist number was decreasing, and so it was very nice things for us to monitoring this situation. But unfortunately, last week, the number of contaminated pharmacists number unfortunately increased, and so we are trying to understand. Maybe the next week and the other week will show us much more the results precisely, because it looks like a second wave maybe, and also it's giving a very good, very hard signal to us. Still, the COVID problem is affecting our lives and our professional lives also. And so we need to care much more maybe than before. Why? Because yes, we learned how to avoid ourselves, but we still don't know what will we do when will we finish this problem. Tonight we will be with Alain Astier. I will introduce him after Klaus Meyer. But today's topic about vaccination, about coronavirus, COVID-19. So it's a very important night for everybody, for all of us. Why? Because we have confused so much, and we have lots of questions in our minds, and I know tonight we will learn lots of things from Professor Astier. Okay, now I want to give the microphone to Klaus Meyer for explaining us the results of our questionnaire and the survey results of the last week. The microphone is yours, Klaus.
Thanks a lot. Thanks a lot. I just want to come back to show you worldwide graph. You see, this is from Worldometer info, what I used to do at the home page, and I will show you a small film. Look at how it's starting. It's starting from the beginning when the first COVID issue was there in March. Then it grew up to April, and now we are at the end of May. Then you see how it has been spread in the world very came from, how it was spread into countries, and you see also then the number of countries who have been influenced a lot. And the issue what Ahmet was talking about the third wave, it's compared, the second wave, it's compared and related to the way that more participants have been online to share their information. And I will go over to this slide then. This is the COVID-19 weekly report we have here, and here you should see the countries which have been participated in Europe. As well, you see these countries mostly have been participated in the past. Sweden came in addition. As well, we got a lots of information from Bosnia and Herzegovina. As well, UK at the first time is this week with us. Then we see Africa and Near East. That's also integrated in Iran, is giving information to us this week. And we see from the Asia, it's white. There's no report. That depends often not only because there are no people who are engaged in oncology pharmacy, but mostly they have probably a different opinion about that to explain the situation where they're working, because sometimes it's not allowed from the state, sometimes it's not allowed by the own mind. And here we see the reason for the big wave this week. We see Brazil came in with a lot of pharmacists reporting to us, and you see there have been really a big number of pharmacies who have been related to contaminated personnel. So when you see that, then you can imagine that the development curve is growing, what is related to the contaminated thing. And I will later on give you a reflection regarding the situation under we are working and the outcome. But you see on this curve what is most important. And I think it's some kind of laziness sometimes to say, okay, I gave my message 4 weeks ago and it should be enough. No, it's not enough. It gives only to us a really good picture when we have a report weekly from everybody, because then we have also a better sight if the changes in this curve are because of incoming new countries or because of the changing situation in all countries. So as we cannot say who has to do it or not, we are doing this anonymously. It's only just a case-by-case research issue. We can find out relations and correlations. Okay, let's go quickly through the report. The first was related then to the number of beds that the hospitals have there, and you see it split it in small and big hospitals as well. You see that they are related also to intensive care beds for the treatment. And you see also that it's mostly hospitals with oncology patients, because these are the hospitals with the biggest number of beds in oncology patients. As well, you see that treatment of outpatient is going up to 50, and the other up to 50 who have more than 20 patients to serve every day. And then the question is how these patients, when they have been contaminated with COVID, how they've been treated. And we have to specialize this question for sure in the next week. You know, we are doing updating every week this questionnaire with questions who are occurring to us to be unclear or better. In this case, we see that the understanding about isolation of patients is different in different countries. Some countries, like in Germany, we have it's when you come in a hospital for different treatment as a broken leg or whatever, you get to be tested. And as long as you have not been declared for uninfected, you are on a ward with all these patients who are in the preview. And the data of the research coming in one day, and then you will be ordered to that ward when you are not infected to the normal ward, or you stay in the ward for a pre-contaminated patient. That means you make a pre-selection in this hospital, and that means that the question of isolated in a general station, if it happens or not. The other question is related to the intensive care for sure, and these are patients who have been hardly influenced by COVID. And then you see this is a growing number of those who have been then going through to the treatment in this case. And one of the most important things is that the chemotherapy preparation decreases over the time. When we take the preparation number in January to 100%, then we see over the time more and more patients get no treatment in the hospitals in their cancer treatment because of different important issues. So that means there's also the question of drug shortage we have discussed, related then to medicaments, to intubation and pain management as well. We have question 12 related to the general shortage in pharmacies as well. Coming to the part of PPEs, you see nearly 60% have a good situation regarding PPEs, but 30% don't have anything in general as well. We see that the governmental issue to support them is not growing after in this short time. Many of the pharmacies have to organize their own equipment, and that means that they have got enough, 60% have got enough, but 29% not. So this means regarding the shortages we have here and delay of distribution, we see that 66% of all pharmacies say they have medicine shortage as well. Coming to the end, we see when we make this relation comparable, then we see that we have the shortage of the 5 medicines who are necessary for the treatment of cancer patients related to medicine shortage in general. And we see that's no degrees of this. It's a growing issue as well. When we see the question, have you got enough PPEs for pharmacists and technicians, and the percentage of affected pharmacy personnel? You see, as long there's not enough PPEs, as there is a result and the reason to have no chance to fight against the infection. So that's why my message in the end, I just want to ask you seriously to talk to everybody you know, to make it happen that in the next 4 weeks, what we have on our plan, in the next 4 weeks to be careful and inform by the data you have in your hospitals in the questionnaire, what is starting every Thursday until Sunday, to give in the data and to make us easily reporting to those who are interested in this. Thanks a lot.
Also, 2 promising developments this morning that we want to get you in the race to find a vaccine for the coronavirus.
To deploy it in every single corner of the world.
The world is waiting for a vaccine.
Scientists are at war with COVID-19. Dozens of laboratories are now battlegrounds in the race for a vaccine.
Okay, after this short video, when Alain is sharing his screen with us, I want to say something about this presentation. First of all, vaccination is so important today. Why, before this webinar, I was looking the results of the total cases and the death numbers of occurred because of the coronavirus, and today's number is 5.5 billion cases, and also 348,000 deaths. So it's a very serious problem for us, and we are locked down in our house and going outside maybe only for buying food and to go to our job because we are working in the hospitals. But still, we haven't seen a light at the end of the road. So it's so important to find a cure. Maybe it will be a medication or it will be a vaccination. And so we have lots of questions in our minds. I hope and I know that you have too. And tonight is a very good night for us. Why? Because we have a very good expert. He is with us tonight, and I know that we will find answers to our questions in our mind. So who is this expert? Most of you, nearly all of you is knowing him, but I want to introduce him to the new ones. Professor Alain Astier get his PhD on organic and structural chemistry and PharmD from University of Paris. I mean, I don't want to read the years when you made your PhD and PharmD, because he made his PhD in 1976. I born in 1975. He's professor on clinical pharmacy and biotechnology in University of Lorraine until 2007. Between 1977 to 2017, he was head of pharmacy department at Harry Moore University Hospital. So 1977 to 2017, nearly 1 century. Member French Academy of Pharmacy and Royal Academy of Medicine of Belgium. He's working on toxicology, pharmacokinetics, new formulations, analyzes, and stability of cancer drugs, especially monoclonal antibodies and biosimilars. He's a very good academician. His research gate score is 41.5. Age index is 32, and number of the citations nearly 4,000. He retired since 2017, and he's presently director of Biotopic Pharmaceuticals, a consulting company in drug development and analysis focused on biologics. And also, the most important thing for us, he is our co-president. He is the co-president of European Society for Oncology Pharmacy. Now, I want to give the microphone and the floor to my old friend, lovely friend, Professor Alain Astier.
Okay, good evening, everybody. It's a pleasure for me to speak to you. And I will start about the topics, which is very complicated, and I will try to explain as better as possible the problem of vaccination. And I will give you, okay. Can you see? Okay.
Everything is fine. If you make a full screen, it will be better.
Okay, full screen will be better. Okay, fine. Okay, the topic is the vaccination against the COVID-2, which is the new one, coronavirus. And the challenge to obtain vaccine against this very bad virus, it's very important, because at the moment we have approximately 6 or 7, depend of the source. We have ensured the clinical trials across the globes, and more than 80, maybe to 100 candidates reported to be in the preclinical states. And you have to remember that there is no current vaccine against another COVID, coronavirus, or cold viruses. And why? Because there is not really an epidemic for these drugs, and the problem is the economic reasons, because the pharma industry had no reasons to develop this kind of viruses. Another problem is that the immune response is very poor. It's a very big problem, because if you have a poor immunogenic response, you can have the poor chance to obtain a very efficient vaccine. And there is some problems with the first vaccine against the COVID-1, which gives some problems in safety in animal models. And the reason is also if we need to develop develop vaccines, because the antivirus drugs seems quite ineffective, such as hydroxychloroquine, remdesivir, and like that. And as is well in medicine, it's better to prevent than to treat. And the idea is to obtain a vaccine. And to obtain a vaccine, the problem is to induce a long-term antibodies. We can destroy the virus if the virus enters the body of the people. And to develop a vaccine, there is two general strategy, traditional methods, and also new generation techniques, such as the mRNA viruses, vaccines against viruses. And we remember that the structure of the coronavirus is very simple. It's RNA, which kills viruses. The RNA of the virus is inside the core of the virus, which is like a sphere. And there is some envelopes containing proteins. And there is two main proteins which are very interesting to develop the vaccine, which is the, and more important, the spike proteins. The spike permits to the virus to enter the cells, the human cells, and to induce the proliferation of the virus. And remember that the basic is the virus enters the bodies because the spike proteins can recognize the ACE2 receptors, which are angiotensin 2 receptors. And in this case, the virus is engulfed inside the cytoplasm of the human cells and inside the vesicles. And these vesicles is destroyed inside the cytoplasm of the human cells and liberates the RNA. And the RNA is activated and can produce the proteins by the ribosomes of the human cells, and it's implicated the replication of the virus. We can be multiplied by 100,000 and billions viral particles which are released outside the cells to contaminate other cells. But the body can fight against the virus by the very important system, which is the immunogenic system, you know, and the virus is ingested by the antigen-presenting cells, APC, and inside these immunogenic cells, there is destruction of the virus, and some part of the proteins of the virus can be presented outside to the T helper cells, and these excitation of the T helper cells induce the proliferation of the B cells, which are very important lymphocytes, which can permit to synthesize the antibodies, which are the ultimate weapons against the virus. Also, the T helper cells can stimulate the cytotoxic T cells. We can kill the cells infected by the virus. But the best way is to stimulate, to promote the synthesis of the antibodies against these virus. And there is several ways to develop vaccines. At the start, you have the virulent parenteral virus, and the idea is to attenuate or to inactivate the virus, to use only the stimulation of immunogenicity, but no bad effect. But we can also fractionate, cut the virus in some parts to obtain a subunit of the virus, and the subunit can be immunogenic, can be induce the reaction of the host against the virus, and also to use some cells, some bacterial cells, to clone the interesting part of the virus to obtain the vaccine. And there is a lot of vaccine used in live attenuated virus, such as for the tuberculosis or for oral polio vaccines, but also inactivated or killed virus, for example, inactivated polio vaccines, but also you can use the purified antigens. It's called subunit of the virus, for example, in hepatitis B cell virus. And also we can use some part of the toxin, for example, which has been secreted by the cells. And for the COVID, there is a lot of possibilities which are actually worked. And for example, the weakened virus is a more conventional way. The idea is to weaken the virus and to inject these attenuated cells, virus cells, inside the human body to produce the replicate of virus, but which are not toxic, but only immunogenic. And in this case, you obtain the production of the monoclonal antibodies, the antibodies, sorry, but without toxic or without clinical effect. But the best is to use inactivated virus, because the problem of to use live virus is they can have a risk of induce the pathologies if the inactivation is not so efficient. And also in sort of patient, in kind of patient, we can be, for example, for immunodeficient patient, the use of vehicle virus, the live virus is not allowed, because there is a risk of induction of pathologies. And the old another techniques is to isolate inside the gene of the spike, for example, proteins of the M proteins of the virus, and to introduce this gene, which we code for the spike protein, for example, which is the immunogenic part of the virus, to introduce this modified DNA inside another virus, which is, for example, the virus of measles, which is not toxic, directly toxic, but we can be a carrier, we can be transport the DNA of the virus inside the human body to induce the proliferation of the proteins and to obtain the immune response. More interesting techniques are the technique using nucleic acid vaccines based. There is two possibilities to use the DNA, like in the preview example, but also to use the RNA. And for the coronavirus, which is the RNA virus, it's a very, very interesting way. The idea is to inject directly to the patients the mRNA of the RNA of the virus, and directly these mRNA will produce the proteins, which is the immunogenic proteins, and which is induce the immune response and the production, for example, of the antibodies. It's one of the best way for the moment, which is developed to obtain a vaccine against coronavirus. And for example, is the approach for one of the small company in the USA, which is Moderna. And the idea is to, like in this slide, the cartoon, you can have an mRNA. The problem is to allow the mRNA to penetrate inside cells, the human cell, and for that we need to encapsule, to create particles, artificial particles, like liposomes, for example, or nanoparticles, and these nanoparticles can be injected inside, for example, the muscle of the human, and in this case, due to the fact that the mRNA is encapsulated inside lipidic vesicles, which mimic the virus, they can enter inside the cytoplasm, and in this case, there is the production from the mRNA by the ribosome of the muscle of the human, the production of a lot of viral protein chain, which can be released outside the muscle cells of the patients, of the peoples, and to induce the formation of the antibodies. It's a very, very clever system, very, very useful, and probably will be the best way to obtain an efficient vaccine against the COVID. If we can prove that this method is efficient in terms of clinical outputs and safety, there is a lot of advantage from this kind of virus. Why? Because it's very simple, very more simple to produce mRNA than to produce virus or cells infected by virus or like that. Because it's only molecular biology. It's simple now with a lot of technologies. We can obtain very, very easily the large production of mRNA if you have the sequence of the virus, which is the case for the COVID-2. And the process is cell-free. We don't need to have cell cultures. You don't manipulate a lot of virus, like for the production, for example, of a virus vaccine, using measles virus or like that. We need to produce a lot of virus. It's very complicated, very expensive, very, very costly, and it's very simple to produce this kind of mRNA. And a paramount advantage is if you have mRNA vaccine with only one scale plant, you can produce a lot of different vaccines. It's very, very simple, very more interesting, for example, for non-profit facilities. And there is also another techniques which can be used. For example, the idea to break the virus, to liberate all the part of the virus, for example, to fractionate the proteins, and to introduce this part of the virus inside an host virus, such as baculovirus. We can be cultivated and produce a lot of poor protein which can be injected directly to the patient, not the mRNA, but the final product with the proteins to the patient to produce the antibodies. But this technique is simple, but the problem is the difficulty to inject to the patients proteins, which can be a problem in terms of safety and easy to injection. Actually, the state of development is very large. We have approximately 100 viruses which are in development, and you have approximately not too much virus directly, but nucleic acid is a large part, for example, but protein-based, which is like in the previous slide from the baculovirus, it's the most important part for the moment, okay? And you can see on this slide that the North America is a more important country to work on the virus, the China, the Europe, and also Australia and other Asia countries such as Korea, for example. And as you can see, the public or non-profit and academic is not so important as compared to the industrial research and production. If you can see here, it's a little bit complicated, but you can have after maybe you can download that, and you can see that there is some strategy with advantage. For example, the mRNA is non-infection, design flexibility, highly scalable, rapid manufacture, non-integrating, but against, there is unstable molecule, mRNA are unstable. We need to specialize administration, the encapsulation inside the liposome, which can be a little bit complicated like that. As an example, if you use attenuated, it's a very efficient because it's potent, induce very strong immunogenic response, and it requires only one single dose. But the problem can be risk of induced disease, especially in patients which are so far cancer patients or like that. Safety testing, which is long time to prove that the product is safe, and for the coronavirus it's not so interesting because we need to have very rapidly a vaccine, and it's also a very large problem to manufacture and store, and they need a lot of very big plant to produce this kind of vaccine. The problem is how to produce billions of those is the problem, and it's easy to develop at the laboratory levels. It's not so complicated to develop a virus, and as you can see, there is 100 or more research, very different kind of virus at university levels, academic levels, or small companies. It's not so complicated to develop a virus. It's complicated to produce the virus, because we need to produce 6 or 7 billion of those, maybe more, if you need to inject 2 or 3 doses per patient to vaccinate the patient. And the problem is the vaccines works, but can we pay? Because if you need to produce very large quantities of vaccine, only very big pharma can produce that. And you can see there is some bitter discussion. For example, the declaration of the director of the Sanofi, which is a French pharmaceutical company, saying that the US will be the first to get potential vaccines created by our companies. And it's a problem. There is also you have to remember that for the polio virus, the Salk, which was the inventor of the virus, didn't patent its virus. It's not the case actually, because you can see at the Wall Street the increase of the output of the company are very, very important. And two key messages: the future vaccine must be accessible worldwide, is very important, and for that you have to reduce the profit of the big pharma, but it's not so easy to do that. Thanks so much.
Thank you so much, Alain, for this nice presentation. It was very informative and very clear presentation. And when you are speaking, I'm trying to collect the questions of the attendees. If you give permission to me, I want to share with you the questions one by one. One question, the starting question is, when do you think will a vaccine be present? What do you think?
I don't know. Sorry, I don't heard you. Could you repeat?
Sorry. When do you think, when will a vaccine be present? When will a vaccine will come to the market, to us? What do you think? Can you give a schedule, a calendar?
It's difficult to say. Normally, for the moment, there is some studies in the USA on humans showing that the product from Moderna, which is the mRNA vaccine, could be efficient in human because there is a very recent publication in Lancet showing that the tentative virus vaccine, from Moderna, which is mRNA vaccine, induced the formation of antibodies. It's a very good answer, a very good response, but it's a very preliminary results, and for the moment, it's too quick to have some maybe in months or maybe 1 year. The problem is the production. Is not to obtain clinical trials, but in clinical trials you will obtain some response. The problem is to produce, and we don't need to have the production of the vaccines in very large scales. Big problems.
Yes, you are right. Mass production will be the biggest problem, the second biggest problem.
Okay. Yes, the biggest problem, and I think if you use the strategy, for example, to use like Sanofi, for example, to use the methyl virus to carriers, the DNA of the coronavirus, it will be very complicated to produce a lot of doses until maybe 2 years, or it's not so good, I think so.
Yes, you're right. Okay, I think that's a very it's an enough answer, and I want to go to the second question. The second question is, our colleague is saying that you are an experienced academician. Doesn't scientific studies of this magnitude require that many experts come together? However, as far as we watch, everyone is trying to find this vaccine in their own country with their own academics. How normal do you think this situation is? Could this be the reason for delay of the producing or finding of the vaccine?
Is the problem of the cooperation. The problem for me is I think there is a very good cooperation for the moment. At the beginning of the problem, we had a good cooperation between the peoples around the structure of the virus, the code, the genetic code of the virus, and like that. But after to produce a vaccine, there is an industrial secret. There is competition between the academic teams, okay? Because there is a good speak about the cooperation, international cooperation, like that, European cooperation, all that. But in practice, in the reality, it's not the same. And if you see the example of the clinical trials for hydroxychloroquines, okay, in Europe, there is our President Macron said, okay, we have a very large clinical trials in Europe with maybe 3,000 or 4,000 people included in the clinical trials like that. In the practice now, there is only 1,000 and mainly French patients and no patient outside. The problem is the difference between the ID you have to push and the reality of the situation. And I think the problem is more linked not to the I come back to my discussion before. The problem is to produce the value, to find the most efficient strategy, not the best strategy. but efficient in terms of the speed to obtain the vaccine is the problem. Because maybe it will be better to have a vaccine which will be not so efficient, okay? For example, requiring 2 or 3 administration or not so efficient. Like, for example, for flu. If you look the vaccine for flu is not so efficient. The vaccine for flu induce maybe a protection of 60, 65, 75% of the population receiving the vaccination are protected against the flu. But it's enough, okay, to stop the epidemics episodes. And also we need to have a speed response. And to obtain a speed response, we need to have strategy which is very rapid, and in terms of production is a problem. It's not the problem to have the best vaccine. The problem is we have to do more rapid vaccine.
Mm-hmm. Yes, you're right. Next question is related by your answer. The question is the passive vaccines need an additional activator. In some countries in the past, with the vaccine against like bird flu, the side effects have been severe, so that people don't want to get a new vaccine again for the bird flu. So what do you think about this?
Yes, the problem is the acceptation of the vaccination is the problem, because there is the more and more people are against vaccines in general. But if you look the poliomyelitis, for example, remember in the 50s and the 60s, there is a lot of people with some very hard pathological problems induced by poliomyelitis, for example. And the poliomyelitis in Europe, it's gone, okay? But the problem is the people don't remember that, okay? And they are against vaccine. Oh, the vaccine is not so good for me. The problem of adjuvant, we could be induce some side effect, but not too much. And it's the problem. And the problem is if you use vaccines which require several administration, this vaccine should be adjuvanted first, okay, and is the problem. And you need to repeat the administration, and is the problem of the number of doses and the problem of acceptance for the patients. But for coronavirus, the problem is the coronavirus attacks mainly old people. And old people are more prone to use vaccination as compared to young people, okay? If you look, for example, in France, the vaccination against the flu, it's free for older people, and there is a very high rate of vaccination in the old people, but not in the young people. But it's not the problem, because the coronavirus don't kill the young people, but kill the old people, okay? And if you have acceptation of vaccination, regardless of side effect or like that, because side effects are minors in general for this kind of vaccine, we will win.
Yeah, nice, right. Intelligent matter was a very intelligent question. Next question is a very intelligent question. One of us asking to you, if you had an infinite budget and talented scientists in your hands, would you spend your resources to find a vaccination for the first time, I think? For the first, would you spend your resources to find a vaccination, or did you spend your money on finding a medicine? Which is the best idea for you?
No medicine. Medicine is not interesting. The prevention is always better than to treat, okay? It's the same thing for cancer. There is no, it's a bad idea to put a lot of money to find new anti-cancer drugs against pulmonary cancer or like that. The best way is to put the money in the prevention. And prevention for this kind of endemic problems is the prevention by using mask, the protection of the face, and also the vaccination. We don't need to have drugs. Drug is too late. Okay, it's a problem, it's too late. If you use the drug, it means that the patient has been contaminated and developed a very severe pathology. Too late. The best way is to put the maximum of money in the prevention in terms of to find vaccine and also to find some, for example, in France, it was very clear. In 2010, we have a very, very large stocks of masks, okay? Half of the whole FFP2 mask in the world was in France, okay? But all is gone now for bureaucracy and like that. It's a problem. If you have the mask, you can prevent, okay? And prevention is always better than the treatment, in my opinion. It's true for any kind of pathology, okay? If you don't smoke, you diminish the risk.
Yeah, you're right. Maybe it started from China, and everybody's thinking that it will stay there, and we will send our sources at first to the Asia, but then we found the problem in our countries. Another question, maybe connected with this answer, as well as it's not a special question about vaccination, but what do you think about the proposals, how to start with a normal life, about using masks no longer, or etc.? What will you see in the future? How will be the normal life?
Oh, no. We cannot imagine to wear a mask for every day in the world. It's not possible. I think it's only the mask should be used in special case, like for the Chinese people, for example. Chinese people having flu wear a mask to avoid the contamination or to avoid to contaminate other peoples. It's the idea. It's the precaution for the mask should be used in some case, but the only way is to develop a virus, okay, is to develop a vaccine. Sorry, because the vaccine will be because look, the problem is, for example, the confinement, the lockdown. Okay, the lockdown. If you look the number of people having antibodies in your blood is very low number. Why? Because the people were too much protected. Okay, in this case, and the best way is to vaccinate.
Yeah. Okay. So one more question. What do you think about how many time that take pharmaceutical formulations development take? About pharmaceutical formulations for the COVID. How many time it will take? Short answers, please, Alain, because we are reaching the end of the session.
Well, the formulation is not the problem, except for the mRNA vaccines, because the mRNA vaccine or DNA vaccine need to be prepared in a specific formulations, which is liposome of like that. The problem to prepare liposome in large scale is not so easy, okay? And it's also complicated to analyze, to control, to produce, to have reproducibility or like that. It's the problem. But the formulation the most simple formulation is to put, for example, proteins. The vaccination using vaccine which is contain proteins is more simple, because you can put like monoclonal antibodies in very simple formulation. But the problem to have a complex formulation such as liposome or nanoparticles is more complicated to produce. It's the limitation. It's one of the limitation of the mRNA virus or DNA virus. We need to have specific pharmaceutical formulation. It's a problem.
Yeah, super. Thank you. I want to say, excuse me to the listeners. Yes, questions are coming, but we are coming to the end of the and our hours. We have a limited time. Only one question I will ask to the Alain, and then unfortunately I want to close the session. Our last and final question, Alain. A little bit long paragraph. It will be a bit of a political question. Please answer if you want. Countries closed their borders, emphasizing the necessity of quarantine. However, we see that just closing your doors, your own doors, doesn't solve the problem. Let's continue on the other side of the world. You have solved the whole problem and open your doors in your own country. It must be why, because it's impossible for a country today to close its doors and stand alone in this century. When you open your own doors, COVID will knock on your door again, because the deaths are still growing in the other side of the world. So the world no longer has to think differently, act like a single world. Is the right thing for you or not? I think the question is I'm trying to understand the last sentence and trying to say everybody closed their borders and trying to solve the problems in their own countries, but maybe I solved all of my problems in my country and I need to open my doors to the world, to other countries, but still another country have the same problem. So why don't we think together or act like a single country, like a single world?
It's a complicated question. The problem is there is an opposition. It's true that if you want to stop the contamination, you need to isolate. It's the problem of lockdown, it's to close the borders. And if you stop to have relationship between the people, you limit the contamination. It's true. But the problem is the benefit of the balance of benefit. If you close all the doors, it's the end of the world, okay? But in terms of, it's like in France, there is some, maybe in Germany, I don't think, but some political people said, okay, you have to close the door of the all peoples until 2021, okay?
Yeah.
From a scientific point of view, very objective point of view, is not so bad. But in terms of social relationship, in terms of life, it's not possible. Okay, there is the opposition between the scientific evidence and the real life, and we must to have an equilibrium between the both. Okay, it's clear. And for that, the best way is the vaccination, because the vaccination, we can stop the contamination, you can limit the epidemics without limiting the relationship between the peoples and to close the borders. Okay, but it's a problem. Klaus, I don't know what is your opinion about that, but it's interesting to see that the Schengen like that, and it disappears like that. There will be, even in between the French and Germany, the border was closed. Okay, it's incredible, no? But it's a problem.
Yes.
I can give you some word.
Okay.
I can give you some word. There's no outcome in the world when everybody's looking at his own. You know, the virus does not look for the border. He's jumping over the border when he wants to do that everywhere, you know, and that's why it's no solution in this unique world to do your own thing. You know, that's for sure, it's clear. And when he stays resistant in parts of the world and you have shut up your doors and the borders, the time will come when he's coming back. So it's not the way to do this in this way. And I think everybody has to look to find out quickly when we are talking about vaccination, quickly the result of it, to go quickly to hand over the people that they can live just a little bit more of the normal life what we are discussing about. I think may I give some words in the end. It was an excellent presentation tonight to see what is evident in the moment and what we can reflect. And I just want to invite you and also invite you to invite others. In 2 weeks, we will have again a webinar with Jürgen Barth, and the main topic is facts and fakes. And you know, Jürgen, he is a great scientific person. He will deliver. He will deliver all resources and preferences for this, that you have a sword in your hand when you fight against stupidity and this what should I say, this idea that something has produced this issue just to make money alone. So thank you very much from my side.
Thank you so much, Klaus. Thank you so much, Alain Astier.
Thank you. Thank you.
It was very nice night, and I want to say a big thanks to the people who choose us for tonight. And I'm closing the session. See you on our next organization of the webiESOP.
Also, two promising developments this morning that we want to get to in the race to find a vaccine for the coronavirus.
To deploy it in every single corner of the world.
The world is waiting for a vaccine.
Scientists are at war with COVID-19. Dozens of laboratories are now battlegrounds in the race for a vaccine.